All right, welcome to Burt's Basics, Box Elder High School Medical Anatomy and Physiology, and get ready for a fun video, but also a beastie video. Today we are doing cell parts. Dear goodness gracious, look at all the words you need to know. Now, I'm going to help you out a little bit. In this class, this is a medical anatomy and physiology class. It's kind of preparing you for the advanced anatomy and physiology that we do at Box Elder High School, which is our Weber State Concurrent Enrollment 8-credit class. This class, you need to know some basic parts and functions. Now, if we could study this, this would take all trimesters to study all these words because they're that important and they can go that much in depth. You need to memorize one key part about each word. One key thing, kind of like when we talk about diseases in this class. If I say one word, say, or two words like clogged oil, you should think acne. If I say twisted, you should think scoliosis. If I say fluid lungs, you should think pneumonia. You got to do the same thing for here. We're going to knock out all these cell parts, and your job is to memorize one key part of each one. So buckle down and get ready to start. Let's have some fun. Let's start with the nucleolus versus DNA and RNA. You know the center of the cell is a nucleus. The nucleus has our genetic material, which we call DNA and RNA. Now this picture on the right looks crazy, okay? But the whole point is DNA is a double-stranded genetic code information that houses the code to build every protein in our entire body. The problem is the parts that make the protein tend to be cruising out here in my cytoplasm. I cannot have my DNA leave. Instead, I make a copy of him into RNA, and then the RNA can leave. So let's see your RNA leaving the nucleus right here? What happens if he gets destroyed here? If he gets destroyed, your DNA is like, seriously? You suck. And then he has to make another copy of himself. As long as my DNA stays intact, we are in good shape. So over here on the left side, the nucleolus is generally the densest region of genetic material inside of a nucleus. That's where the DNA is making all of its fantastic stuff. Now, if you're going to make stuff, you need to know what part of the DNA is making stuff. Our DNA goes by lots of names, okay? When it's in its loose states, we call it chromatin. When it's in its coiled up state, getting ready to divide, we call it a chromosome. But I don't care about those words as much as I care about this word right here, which is a gene. A gene is a segment of DNA that codes for something specific. So let's say this is the gene right here for insulin. That means that code of DNA houses the blueprint on how to make insulin. That's called a gene. Now, if we get a mutation in a non-gene region, it's not very scary. If you get a mutation in a gene region, that can be pretty scary. Okay? Now let's talk about who's going to be reading that. Ribosomes. Now, let's start basic and then let's go complicated. Basic. Ribosomes make protein. Ribosomes make protein. Ribosomes make protein. Ribosomes make protein. Ribosomes make, what are they making? Oh, that's right. They make protein. Okay? That's probably the test question to everything you're going to have in this lifetime is ribosomes make protein. Now, let's go a little more AP now. In order to make a protein, they have to read RNA. Now, this RNA is coming from the nucleus. It's a copy of a gene. And then they have another form of RNA who's bringing these blue beads. Now, if we look at these blue beads are amino acids. So this tRNA is just bringing the amino acids to the party. And then I grow this polypeptide chain, which you guys know is an amino acid. So remember how we talked about this is the gene for insulin? That means that this blue guy coming out is insulin. So the question you should be asking is, man, how come ribosomes are so good at grabbing RNA and then making a protein? Now it's time for AP. This is another form of RNA. It's called ribosomal RNA or rRNA. Ribosomes are made in my nucleolus. All these guys are made in my nucleolus. So let's take a peek. If I want to make a protein, I need a ribosome, made in the nucleus. I need my RNA copy of the gene, made in the nucleus. I need my taxi cab driver who's bringing the amino acid to the parties, made in the nucleus. So my nucleus literally is in charge of every single protein in my body. That also means that if you are lacking protein, you need to crank that sucker up. The nucleus can pop out 50,000 ribosomes at the same time. I mean, it's amazing how good our body is at homeostasis. Now let's talk about ribosomes. Most of the time, they can be floating in the cytoplasm, but they tend most of their life to be anchored to somebody. And that's where ER comes into play. Okay. Now in class, we're going to talk a little bit about this in depth. We're going to talk about the actual mechanism of rough versus smooth. But remember, this is Burt's Basics, so let's talk about it. The ER first. Okay. ER. All this in blue is ER. Now, there's two different types of ER. There's those that lack ribosomes, which we call smooth. There's those that have ribosomes, which we call rough. So there's a pretty good difference between rough and smooth. And then there's my nucleus. Okay, the membrane around the nucleus is called your nuclear envelope. Okay, those are just where everything is in. Now let's go to this class. It helps if you knew what ER did. ER really focuses all day making lipids. It is the lipid master. It can make steroids and lots of different things. It makes lipids most of the day. Most books also say it's very good at synthesizing carbohydrates or altering carbohydrates. So I teach it simply as the ER can do two things: make lipids and carbohydrates. Now guess what happens if you add ribosomes on there? Remember, ribosomes do one thing and one thing only. They make protein. So if you're talking about rough ER spends most of his day making protein. If you're talking about smooth ER spends most of his day making lipids and synthesizing carbohydrates. But if you put those together, you know that nucleic acid is just made of the three. It's made out of carbs, lipids, and proteins. That's why the ER surrounds the nucleus on all sides. So on the test question, if I say true or false, ER makes protein, that is false. But if I switch it to rough ER makes protein, that now is true, because when you say rough, it now has the ribosomes there. I guarantee that's going to be a test question, so get ready for it. It's going to be tricky, okay? Once ER makes its products, it's going to send its products out to the Golgi apparatus. Now, the Golgi apparatus, you've learned in sophomore biology, is the packaging center or the UPS center. In class, once again, we're going to go a lot more advanced in this, but I'm just trying to do the basic concepts in this video. The vesicles will come to the Golgi, and the Golgi, in humans, it's generally between 4 to 6 of these discs called cisternae, okay? And what they do is they open it up, and they make sure the vesicle or the bubble has everything that it needs to have. And if it does, it lets it keep going through. If it doesn't, it will hold it and sort it and add to and take away. When this bubble pops out the other side, it is completely modified, perfect, and has everything in the world that it needs. And we call these bubbles popping out the other side vesicles. And there's only one vesicle that I'm going to test you on, and it's a very specific vesicle. The Golgi will take a bunch of enzymes. Now, enzymes are all proteins. They're made by ribosomes or rough ER, and they all end in ACE. Most end in ACE, okay? And so I get this bubble, and I throw in all these digestive ninjas, and then I'm like, okay, you bubble, anything comes to my cell, you're going to kick its butt. And then that poops it out the vesicle on the other side. This vesicle is called the almighty lysosome. Now, in med term, some means body, right? So that's your body. Lyso in med term, I think, means to dissolve. I never teach it that way. I teach it as lysos to rip your face off. Okay, so a lysosome is a body is going to go around and rip your face off. So let's look at our drawing. Here's your Golgi apparatus. Here's all the vesicles it's getting out. It's this bubble right here. Now let's look. Who made the lysosome? The Golgi did. Who made the enzymes in the lysosome? That would be ribosomes or rough ER. And all these dots right here represent enzymes. So in this case, I have food. I'm doing active transport, which we saw in our last video. Phagocytosis, pulling the food in. I now have a vesicle. Look at these lysosomes merge. And then all of a sudden, these enzymes start to destroy the food. Once I destroy the food, I will use diffusion and osmosis and facilitate diffusion to get all the stuff out of it I want, and exocytosis after. Lysosomes are awesome. They destroy a lot of cool things. Okay? Mitochondria. In USU biology, we spend a whole 2 weeks on mitochondria. In this class, we're focusing on one of its concepts and only one of its concepts, which is if you take fats, lipids, and you take sugars, carbohydrates, and you send them to the mitochondria, they have all these cool features they can do to metabolize them and transfer the energy into the almighty, all-powerful ATP. If you see ATP on the test, it's going to be mitochondria is your answer 100% of the time. Okay, in sophomore biology, you learned it as the powerhouse, okay? That's another word to say it. Cytoskeleton, the forgotten child of a cell. In class, I'll probably rant about this for half an hour because I really think this is more important than anyone gives it credit for. But the cytoskeleton is a series of protein sticks called microtubules, microfilaments, okay, that give the shape of the cell. They give it structure in a three-dimensional object. They also play a huge role in cell transport, which we'll talk about in class with dying and walking proteins. But in our class, right now, we're just doing the basic structure of a cell. You guys are rocking. Cilia versus flagella. Now, if you look at them, they both look like tails, okay? The difference is cilia, many. Flagella, one. Cilia, shorter. Flagella, longer. Cilia on the surface of the cell helps to move things across the cell to give it a little bit more surface area, okay? Flagella, main purpose, to move the cell to where it needs to be. In the human body, there's only one cell that has a flagella, and that's the sperm. Okay, in a human body, there's a lot of places that have cilia. Most common is your respiratory tract. Your mucus gets moved up by your cilia, up, 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 up into your throat. You know, you've all been sitting in class and you do that cough where you're like, huh, and all of a sudden a loogie comes in your mouth and you're like, oh, okay, that's the cilia doing its job. Okay, it's getting the mucus out of your respiratory tract and into a different system. Okay, so that's cilia versus flagellus. These are hair-like, but don't call them necessarily hair because they're not hair, okay? So cilia help move things across the surface. Flagella helps move the cell itself. The last thing you need to know is mitosis versus meiosis. Once again, in USU biology, we spent almost 2 weeks on this. This is a huge concept. Oops. Okay, well this says, let me pause this for a second. There you go, now you can see the diagram a little bit better. When I first saw mitosis and meiosis on the medical anatomy core, I was like, holy shnikes, because I was thinking prophase, metaphase, anaphase, telophase, cytokinesis, and all the steps in between. But from my experience teaching medical anatomy, they just want you to know the 2 words and what is the end result. We have 2 types of cell division. So if we take any cell, this one has 4 chromosomes. Can you see 1, 2, 3, and 4? Our human cells have 46, okay? So this one only has 4. If you do the almighty mitosis, it will go through 1 round of cell division and you end with 2 cells. Now look, these cells also have 1, 2, 3, and 4. Genetically identical to the parents. So if I hurt my skin and cut my skin, I have to do mitosis to make identical skin cells. If my liver lacerates and I need to replace some of the liver, I need to do mitosis. You do mitosis in 99% of your body. If your eye gets damaged, mitosis. If your muscles get damaged, it's mitosis. Everything is mitosis. The goal is you want to make 2 identical cells. Meiosis, on the other hand, is different. Now, I pronounce it meiosis to make it sound different. Most teachers would pronounce it meiosis, okay? In meiosis, you don't do 1 cell division, you do 2. And notice you end with 1, 2, 3, 4 cells instead of 2. And if you notice, the original cell had 4. The daughter cells have 2, so these aren't even full cells. These are half cells. In the world of biology, we call these gametes or haploids or anything like that. These are only done in gonads, which are testes and ovaries. These are the makers of sperm and egg. So on the test, meiosis is sperm and egg, half cells, gametes, okay? Only in ovaries and testes, nowhere else in the body. Mitosis is generally when I'm repairing cell damage or tissue damage, and I want to make identical cells between the 2. Congratulations, you were amazing. You knocked them all out. So here's your review. Your review is the following. Look at this diagram and do the best you can. Label every letter. Not only label every letter, put what they do, and then you'll be ready for the test. Some of these are going to be a little harder than others. The answer key is coming right up. I hope you enjoyed Burt's Basics, and I'll see you soon. Let me move my face so you can see the answer key. There you go. I hope you rocked it.